It's Fact or Fiction with your host, Lucy.
The story of the historical development of cholesterol as a heart health risk factor spans over a century, evolving from early animal experiments to genetic discoveries and life-saving pharmaceutical treatments. No subject relating to human health has received as much attention or provoked as much discussion as cholesterol. Today, it is centered at the crossroads of the pathway to CVD. Here, briefly, is that story. In this short video, the first in the Cholesterol Story series, we follow the story of cholesterol from its first discovery and naming in the mid-20th century to the 1950s when the scientific groundwork was laid for what later became the diet-heart hypothesis, a period declared as the cholesterol era by renowned lipid scientists Goldstein and Brown. Here, we scroll through the timeline of key points so that we can see the overview and understand the context of each major milestone on the journey to the diet-heart hypothesis. The transition from a mysterious waxy substance in gallstones to a named chemical compound was driven by two French scientists, François Pelletier and Michel-Eugène Chevreul. François Pelletier de la Salle, 1769, was the first to isolate cholesterol in its solid form when he extracted shiny crystals from the alcohol-soluble part of human gallstones. Because his work was never officially published, his contribution is mainly known through quotes by contemporaries. Michel-Eugène Chevreul, 1815, rediscovered the substance and coined the name cholesterine. The name comes from the Greek chole, bile, and stereos, solid. He proved that the substance could not be saponified, turned into soap, distinguishing it from common fats and waxes. Known as the father of lipid chemistry, he also identified cholesterol in both human and bovine bile in 1824. The name was later updated to cholesterol in the late 19th century after scientists identified the OL suffix was more appropriate for its chemical structure as an alcohol. Rudolf Virchow, 1856, shifted the focus of cholesterol research from a chemical curiosity to a primary cause of disease by identifying its role in the development of arterial plaques. Virchow was the first to describe lipid accumulation within the arterial walls of individuals who died from vascular diseases. He proposed that atherosclerosis was not a passive buildup of fat, but an active inflammatory process, and that the thickening of arterial walls was a cellular reaction to chronic irritation. He formally described the atherosclerotic plaque as the fundamental lesion of the disease, noting that it formed within or beneath the inner, intimal layer of the blood vessels. He correctly identified that this fatty metamorphosis led to the narrowing of arteries, which decreased blood flow and increased the risk of heart attacks and strokes. While broader than just cholesterol, he defined the three causative factors that predispose a person to thrombosis, a framework still used by the National Library of Medicine to understand how plaques lead to clots. His inflammatory hypothesis was so advanced that it was largely overshadowed by simpler, passive fat theories for over a century, only to be fully validated by modern medicine in the late 20th century. In 1910, German chemist Adolf Windaus, who later won the 1928 Nobel Prize in Chemistry, provided some of the earliest scientific evidence linking cholesterol to cardiovascular disease. He published a landmark study showing that atherosclerotic plaques in human aortas contained significantly more cholesterol than healthy arterial walls. His research specifically identified that these plaques contained roughly 20 times more esterified cholesterol, the storage form, and 6 times more free cholesterol, the base form, compared to a normal arterial wall. This discovery was a first hint that the cheesy substance, atheroma, found in clogged arteries was composed of lipids. It laid the foundation for the lipid hypothesis, which suggests that elevated plasma cholesterol levels are a primary cause of coronary heart disease. While Windaus's work was purely observational, conducted through autopsies, it shifted the scientific understanding of atherosclerosis from a simple consequence of aging to a condition potentially driven by metabolic or dietary factors. Russian pathologist Nikolai Anichkov, 1913, provided the first experimental proof that cholesterol alone could cause arterial damage by feeding rabbits pure cholesterol dissolved in sunflower oil. He proposed that atherosclerosis begins when cholesterol from the blood infiltrates the arterial wall, rather than being a result of local inflammation or aging alone. Anichkov was the first to identify and describe foam cells, lipid-laden macrophages that are the hallmark of atherosclerotic plaque. He meticulously documented the life cycle of a plaque, distinguishing between early fatty streaks and advanced atheromatous plaques. His long-term studies showed that if cholesterol was removed from the diet, early arterial lesions could actually regress, a concept that remains central to modern preventative cardiology. Despite publishing in top German journals, Anichkov's findings were dismissed by many because researchers could not replicate them in dogs or rats. It was not understood until much later that these animals are far more efficient than rabbits or humans at converting dietary cholesterol into bile acids. Rudolf Schonheimer, 1933, fundamentally changed our understanding of cholesterol from a static substance to a dynamic, internally regulated system, using deuterium oxide, heavy water labeling, a technique he pioneered. He proved that the body doesn't just get cholesterol from food, it manufactures its own cholesterol from smaller molecules, like acetate, and he discovered a thermostat effect, whereby when an animal eats more cholesterol, its body slows down internal production to maintain a balance, and he identified that the primary way the body gets rid of excess cholesterol is by converting it into bile acids and excreting it through the gut. He also overturned the idea that body fat and cholesterol are permanent storage by showing they are in a constant state of rapid turnover, being broken down and rebuilt daily. His work explained why some people can have high cholesterol despite a low-fat diet. Their internal factory is simply processing it in a different way. In 1938, Carl Miller provided the first definitive evidence that high cholesterol and heart disease could be written into a person's DNA. His study of Norwegian families transformed cholesterol from a lifestyle issue into a hereditary condition. Muller was the first to describe FH as a specific inherited disorder. He noticed that in certain families, multiple members suffered from extremely high cholesterol and heart attacks at very young ages. By tracing family trees, he demonstrated that this condition is passed down as a dominant trait, meaning a child has a 50% chance of inheriting it if just one parent has the gene. He identified a clinical pattern that doctors still look for today: hypercholesterolemia, high blood cholesterol, xanthomas, yellowish cholesterol deposits on tendons or eyelids, and angina pectoris, chest pain from heart disease. Muller's work was crucial because it showed that heart attacks in young, otherwise healthy people weren't bad luck. They were the direct result of a lifelong, genetically driven elevation of cholesterol. Miller's clinical observations laid the exact groundwork that Brown and Goldstein used decades later to discover the LDL receptor, which eventually won them the Nobel Prize. Dr. John Goffman transitioned the cholesterol story from animal experiments to human clinical reality in the late 1940s and 1950s. Known as the father of clinical lipidology, his primary contribution was proving that cholesterol does not float freely in the blood, but is carried by specific particles called lipoproteins. Using an analytical ultracentrifuge, Goffman developed a method to separate blood fats by density. He was the first to identify and name the major classes of lipoproteins we talk about today: low-density lipoprotein, LDL, and high-density lipoprotein, HDL. By studying heart attack survivors, Goffman proved that they had significantly higher levels of LDL than healthy individuals. This finally confirmed that Anichkov's rabbit findings applied directly to human heart disease. He also noted that higher levels of HDL appeared to be protective against coronary heart disease, laying the groundwork for our modern understanding of good versus bad cholesterol. And he introduced the atherogenic index, a way to calculate a person's risk of a heart attack based on the specific balance of these different lipoproteins in their blood. Goffman's work was so revolutionary that it was initially rejected by major medical journals, yet it led to the Framingham Heart Study, which turned these lab findings into public health advice for everyone and ultimately became the foundation for every cholesterol blood test used in doctors' offices today. Looking back, we can now see how decades of scientific research conducted last century shaped the discussion and regulations around dietary cholesterol and how the fundamental hypotheses of last century lingered into the present, setting the stage for the global war on cholesterol that was yet to come.